Law of segregation or monohybrid cross. The phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. The Mendel law of segregation is a law of inheritance proposed by Mendel in 1866. According to this law, each organism is formed of a bundle of characters, each character controlled by a pair of genes or factors. During the gamete formation, the two factors of characters separate and enter different gametes and enter into different gametes. This law is also called law of purity of gametes. Law of purity of gametes. The monohybrid experiment explains law of segregation. The crossing of two plants differing one character is called monohybrid experiment. The crossing of two plants differing in one character is called monohybrid experiment. See, here it is the monohybrid experiment table. The monohybrid experiment is also called law of segregation. According to the law of segregation, the parental plants, Mendel carried out monohybrid experiment on pea plants, and based on the results, the monohybrid experiment, he formulated the law of segregation. Mendel selected two pea plants. The two pea plants are one with a tall stem, another one with a dwarf stem. Tall stem is the dominant character, dwarf stem is the recessive character. Dominant character plant, parental plant containing capital T alleles of genes, and recessive parental plant, dwarf parental plant contains small t homozygotic genes, alleles. They are in homozygotic condition. The dominant parental pea plant and recessive parental pea plants are in homozygotic condition. The dominant parental pea plant contains capital T genes, and recessive pea parental plant containing small t homozygous genes. These two parental plants, from these two parental plants, one parental plant act as a male and one parental plant act as a female. By the emulsification, anthers are artificially removed from the female flower, called emasculation. By the emasculation, parental plants act as one parental plant act as dominant, one parental plant act as a recessive female, male and female plants. Artificially, sexual organs are removed from the parental plants. For example, we take male plant containing only anthers, called male plant. Female plant contains ovary, called female plants. From the female plants, artificially anthers are removed. From the male plants, artificially ovaries are removed, called emasculation. These two male and female parental plants are crossed to produce capital T gamete from the dominant pea plant, small t from the recessive pea plant. These two gametes are crossed to form F1 generation, filial generation 1. The filial generation 1 express only tall characters, but The filial generation 1 plants are in heterozygotic condition. The heterozygotic condition plant P plants containing capital T small t genes. This position is called a heterozygotic condition. The capital T small t filial generation 1 P plant express only tall character, tall stem character, because the dominant gene is present on the filial generation 1. So it express only tall character, tall stem character. The tall stem character of filial generation 1s are self-fertilized, are self-crossed to form F2 generation, filial generation 2. In filial generation 2, the phenotypic ratio is 3:1. The phenotypic ratio is 3:1. 3:1 means the phenotypic ratio means we identified with the naked eye. The characters or colors are identified with naked eye called phenotypic characters. In F2 generation, the monohybrid ratio, phenotypic ratio is 3:1. 3 means tall plants and 1 is dwarf plant. See here it is, from these tall plants, homozygotic tall plant is 1, capital T, and heterozygotic tall plants are 2, capital T, capital T, small t. So these 3 plants are express tall stem character, tall stem character. So these are 3 and 1 is tall, 1 is short, called dwarf. It containing only small t genes. The monohybrid phenotypic ratio is 3:1 and monohybrid genotypic ratio is 1:2:1. It explained with Punnett checkerboard. See children, it is the male plant gametes and these are the female plants gametes. The male plant gametes capital T small t, and similar the female plant containing capital T small t gametes. They are fused capital T. It is homozygotic tall plant, and capital T fused with small t. Another E capital T small t. Capital T express homozygotic tall character. Capital T small t express heterozygotic tall character, means these 3 are belongs to tall stem character, and the last one is small t. The character is dwarf stem. So genetically we identified 3 types. One is 1:3:1. 1 means homozygotic tall plants, 2 means heterozygotic tall plants, 1 means homozygotic dwarf plants. Genetically, the monohybrid ratio is 1:2:1 and the phenotypic ratio is 3:1. It is the law of segregation or monohybrid cross.